Thank you for joining the symposium. I will just introduce the reason why we are here. And this is a symposium organized as a part of the strategic partnership between the University of Lausanne and the University of Padova within the context of a collaboration between the Psychophysics and Neural Dynamics Lab and the NeuroMove Rehab Lab in Padova. I take this chance to thank already the Alemanic Neuroscience Doctoral School and the Sense Innovation and Research Center for their for sponsoring and supporting this event. And let me introduce why we are here, what we do in terms of our research at the Psychophysics and Neural Dynamics Lab. We are interested in studying human perception and cognition using a combination of techniques that goes from behavioral methods like psychophysics to computational modeling and neuroimaging techniques, particularly electroencephalography. And at the NeuroMove Rehab Lab, from which we have Maria here, they are instead interested in developing personalized rehabilitation protocol, integrating approach based on neurophysiology and movement analysis to enhance the quality of life of individuals with pathological conditions. So, as you can see, these two fields are not completely different, but not really closely related to each other. So why are we here and why we are building this collaboration? This starts from our common shared interest in using electroencephalography, EEG, as a core tool in our research. Whether it is about studying the neural dynamics underlying our perception or our cognition doing a visual task, or studying processes like motor control and proprioception. And the idea is to bridge our knowledge and integrate findings from EEG research on visual cognition and motor control. For those of you who are completely new to this technique, I just spend a few words. So EEG measures mostly postsynaptic potentials generated by large populations of neurons firing together in synchrony. And this population activity can be recorded, propagates through several layers of tissues, and can be recorded from the scalp with an electrode, for example. And the scalp EEG has the advantage that it's a non-invasive technique, so it is of no harm for the subject. And also the clear strength, that is a method with high temporal resolution that allows us to take snapshot of neural activity at the sub-millisecond scale that is relevant, for example, if we want to measure the neural dynamics that support the execution of a movement or the perception of an image. And the history of EEG is also a fascinating one. It is now 100 years since Hans Berger recorded his first EEG. Berger was driven by strong dedication and I would say ambitious aims. And he spent years in trying to develop a method to record electrical activity from the scalp. Yet, after he obtained his first results, he remained highly skeptical of his own findings, taking additional years before sharing them with the community. And I can only imagine his thoughts after obtaining his first EEG recording that you can see here, as he observed this regular pattern in brain activity, basically oscillations at around 10 Hz, 10 cycles per second, now known as the alpha rhythm. And I can imagine he was questioning, was this a groundbreaking discovery or simply an artifact, something of no importance? And moments like this are very rare in science. And I guess little could he have expected that 100 years after this very moment, hundreds of researchers worldwide would gather to celebrate the profound contribution of the EEG in our understanding of the human brain and behavior. The history to arrive here was not without challenges. the history of EEG research was marked by ups and downs, much like the periodic nature of this brainwave. Initially, for example, this alpha activity, which is mostly observed when we are in a resting state with our eyes closed, disengaging our attention from the world, and I hope you will not have too many of these states today, was taken with some skepticism and considered as something related to resting state that may be not relevant to understand active behavior and cognition. But as you will see, this view has changed drastically over the years. There have been many different theories and new findings. And now we consider alpha activity and brain oscillations in general as a fundamental aspect of brain functioning. And it's a key feature in both in basic and clinical research. Despite this progress, there are still open questions, which makes the EEG research a continuously evolving, dynamic, and engaging field. And the aim of today's symposium is exactly to present you with some of the most recent challenges, theories, and view on the EEG by promoting the research of early career researchers who dedicated their PhD, postdoc, and research activity to the study of EEG. And the symposium is structured into 3 different parts in which we will go through novel computation and modeling approach. We will discuss some important methodological considerations, and in the end, we will have several talks showing application of EEG to the study of perception and cognition. There are 8 researchers. The last 2 will be in Zoom because they are from the US, and I hope you will enjoy the symposium.
Okay, thank you, David, for the introduction. So we will start with the morning section. So as David has already spoiled, so the morning section is about novel computational and modeling tools to investigate the functional role of brain oscillation and their potential application in clinical settings. So here is the schedule of this morning. We will have 3 talks. The first one from Andrea Alamia, that would be about the predictive coding perspective on oscillatory traveling waves. Then we will follow with the Isotta Rigoni about the EEG brain networks for epilepsy application. And then we'll talk from Edoardo Passarotto about the sensory motor adaptation in rehabilitation. Okay, mainly all these talks are applied to clinical settings. So first, we will have some insights and application of this predictive coding in a schizophrenic patient, I suppose. Then we will go on with this special study also of brain oscillation, because Isotta will present us the graph signal processing, coupling structure and function connectivity. And then Edoardo will state how much is important in understanding the EEG correlates to understand also the behavior and study the behavior of our patient, in particular during and after rehabilitation. So here are our 3 speakers with their first 3 teams of our morning. So we will start from Andrea Alamia, and then I will introduce him. So Andrea obtained his PhD from the Université Catholique de Leuven in Belgium, working with Alexandre Zenon and Etienne Olivier on unconscious learning. He then moved to Toulouse for a postdoc with Rufen Verhullen, working on neural oscillation and predictive coding. After a summer internship at the Reich Institute in Japan, he did a second postdoc between Toulouse and the Brown University, investigating brain-inspired machine learning models with Rufen Verhullen and Thomas Serre. Currently, he holds a CNRS position in Toulouse, where he's carrying on an ERC starting grant investigating the role of traveling waves in predicting coding processes. So I will leave the stage to Andrea.
All right, so thank you everybody for being here. Thank you so much, Maria, for the kind introduction, and Maria and David for putting this together. It's really great to be physically in Lausanne, so thank you so much for having me. So today I'm going to talk indeed about predictive coding and traveling wave. So let me remind you in a nutshell about predictive coding. So the idea is that sensory system, like the visual system, are generative hierarchical processes, where higher region predicts the activity of lower region. And the difference between the prediction and the actual activity is the prediction error, which is used to update the next prediction. So in other words, the idea is that the brain or the visual system has a model of the world that allow us to that generation about the environment, and so allow us to efficiently interact with our surroundings. So during my postdoc with Rufan, we developed a very simple model that does basically exactly the same thing. Then, I don't know if you see the mouse. No, you don't. Okay, so basically, we develop a simple model of predictive coding where two-level model, where the higher level generates prediction YDT to predict the activity of the lower level, level 1, so the input. And the difference between the prediction and the input is the prediction error, which is actually used to update the next prediction. So a very simple predictive coding model, but we also introduce biological constraints. So for example, the temporal delay, it takes some time for the information to propagate between levels in the system, and also time constant, the tau and tau d. And what we observe with this very simple minimal implementation of predictive coding is that if we consider, for example, the prediction YDT as a proxy for our EEG signal, then we see that this system, very simple minimal predictive coding system, has an oscillatory behavior. So its impulse response function that characterizes how the system responds to impulse, so to an input, is actually an oscillatory dynamic. So it's oscillating over time. And specifically, this oscillation is depends, the frequency of the oscillation depends on the parameter of the model. So delta t on the x-axis and tau on the y-axis. So you can see that if we take biologically plausible value for delta t and tau, so around dozens of milliseconds, which is roughly the times that it takes for the information to travel between, let's say, V1 and V2, then we are in this region in red, which is the alpha band. And interestingly, if we shorten this delay, so if you have a more local dynamics, so more shortened time scale, so 5-10 milliseconds, then the frequency became higher, which again, it kind of makes sense intuitively. So the more local dynamics, the faster the dynamic, the higher the frequency. So the first very simple take-home message is that a very simple predictive coding model with minimal implementation and biological constraints give rise to oscillatory behaviors. Then we had fun to expand the model to make it a bit deeper and have n layer. And the equation look a bit scarier, but it's exactly the same thing. So the prediction is just updated with the prediction error, which is a difference between the prediction and the actual activity. And we also introduced And we also introduced at the very high level a prior. So there is some prior activity that keep generating prediction about the environment. So the input was just white noise, so just some random number that we fed to the model, and the prior matched the statistic of the input. So it was also white noise. And what we observed is two things. The first one, so on the left part, you see on the x-axis the time, on the y-axis the layer, and yellow and blue are the peak and trough of the oscillatory activity of all layers. So we first see that each region, each layer, the activity of each layer is oscillatory. So again, we confirm the first idea in the first model. So predictive coding, even with simple implementation of predictive coding with temporal delays, generate oscillatory dynamics, even if we expand the model. And more interestingly, this oscillatory activity propagates as a traveling wave forward or backward, depending on the cognitive state of the system, which means that if we feed the input, if we feed the model with the input, so if we provide some white noise to our model, then this input generates some oscillatory activity that propagates from the lower part all the way up to the higher level. If we don't give the input to the model, then the prior activity takes over and propagates from the 7th layer all the way down to the lower layer. So a very simple predictive coding implementation generates oscillatory dynamics that propagate forward or backward depending on the cognitive state of the model. So if you give an input or not. So this is a very simple implementation. I would like to mention very briefly the work that Jakob Schwenk did in very recently. Actually, he basically take the same model and make it more biologically plausible. So each region, originally the model was very simple. He developed a model in which now there are some cortical dynamics with laminar dynamics within each region. I won't enter into the detail, just to say that the very simple Qt model that we developed originally can actually be adapted to a much more biologically interesting and plausible interpretation. All right, so very nice. We have a cool model that can generate oscillation that propagates forward and backwards, but what about real data? How can we actually match and validate the hypothesis that our model generates? So we look at EEG data and we focus on the midline electrodes. So from OZ all the way up to FZ. So we take the midline electrodes and we basically recreate the 2D maps that we saw before. So on the x-axis we have time, on the y-axis we have the 7 electrodes, and once again the color represents the activity of each electrode. So the yellow are the peak, And in blue, you have the trough of our oscillations. And you can see already with naked eyes that there is this oscillation which starting from occipital electrode and propagates somehow all the way up to a frontal region. So this is a schematics fake data, simulated data to show even better how we think of oscillation in EG in these lines of electrode. So you have some pink noise activity, then an oscillation starts, let's say occipitally, and there is a constant phase shift that propagates all the way up to the frontal electrodes. And this is a forward oscillation, a forward traveling wave. Or vice versa, the activity could start in the frontally, so in the first electrode, and propagate all the way down to occipital electrode. And this is a backward traveling wave. Okay, so we have forward wave, we have backward wave, we can see them somehow in our EG. How do we actually quantify them? So we'll go very quickly on this, just to give you though a sense of all the data that I'm going to show in the next slides. So we basically create our 2D maps. We have a sliding time window. So on the upper part, you can see the EG signal from OZ to FCZ, so our midline of electrodes. We have a sliding time window that we make run through the signal. And from this slide window, we again extract these 2D maps with time and electrodes on the y-axis. And from here, we apply a 2D FFT, which basically tells us in the upper quadrant, it's a bit annoying that I don't really have a cursor. So in the upper quadrant, it basically tells us how much power there is as a function of the frequency of the oscillation propagating forward. And the lower quadrant tells us how much power is there in the wave propagating backward. So we have a measure of forward and backward wave as a function of the frequency. Now we need a baseline somehow. And to do this, we basically take the same data, we shuffle the order of the electrode, so we destroy the spatial information, and we apply the same 2D FFT. So basically now we have a surrogate distribution, so some surrogate or null distribution power, forward SS and backward SS, that we use as a baseline. So we basically have destroyed the spatial information and kept only the temporal one as a sort of null distribution. And so all the data that you will see from now on, they are expressed in log ratio. So it's basically a ratio between the real wave, the order, let's say, data, so with the electrodes in the right position, compared to the shuffle one. So if this number is around 0, it means that there are no more waves than random. If I shuffle the electrodes, I get the same amount of waves. If this number is bigger than 0, then I have more waves than chance. All right, so now we have our method to quantify traveling wave. Well, let's see whether we can see these waves in actual EG data. So I'm going to present some work that was done by Tso-Yang Pan, who was a PhD student in the lab. And what she did basically was trying to understand whether we can see this forward and backward alpha traveling wave during visual stimulation. So she presented the participant with 5 seconds of visual stimulation, either a very dynamic flickering or a very boring static 5 seconds of white disk. And these 5 seconds of visual stimulation were alternated with 5 seconds of blank screen. And the room was very dim and sleepy somehow. And so what she did, she computed this traveling wave during visual stimulation in the dynamic or static condition and during the rest. And what she found is very much in line with the prediction of the model. So on the x-axis, you see the time. On the y-axis, the amount of waves in decibel. And blue and red are the forward blue and the backward, the red waves. So during when there is no visual stimulation, so the white part here, then there are a lot of backward wave and not forward wave, as predicted somehow by our model. Whereas when we have the visual stimulations, the shaded area, then there is an increase of forward wave and a decrease of backward wave. Again, very much in line with the prediction of our model. So visual stimulation generates wave propagating forward, lack of visual stimulation generates. Then the backward wave take over and you see them more clearly. And this is true for both the dynamic and the static condition. All right, so somehow the prediction of our model was somewhat validated. But then if we really commit to this predictive coding idea that backward wave carries prediction and bottom-up wave, alpha wave, carry prediction error or sensory information, then we can push our interpretation a bit farther and we can generate more specific hypotheses. So let's imagine that we are driving a car and we are estimating the distance of the car in front of us. So if it's a very sunny day because we are, I don't know, in Madrid or in Toulouse, it's summer, it's sunny, we are not high, we didn't drink anything. So the car in front of us is, let's say, 3 meters plus minus 10 centimeters. So we are in the right case. So we have a precise estimate of where is the car in front of us. So in this case, in theory, according to our predictive coding interpretation, we would have more top-down prediction and very little prediction error. We don't need to update our model of where the car is because we can still predict it efficiently. On the other end, if you are driving in Glasgow and it's rainy, foggy, and maybe we had a few whiskey because Scotland, why not? then the car is 3 meters in front of us, plus minus 1 meter, but maybe too much, but plus minus larger distance. So in this case, we are on the left case. So we have relatively little prediction and much more prediction error or sensory information. We need to keep updating how far is the car in front of us because we want to make sure we don't crash into it. And so if somehow we manage to be in these two conditions experimentally, we could test whether we can see more backward or forward wave in these two conditions. And that's what we did kind of indirectly, but I like to show it anyway. So on the first part was the first experiment that we did was a collaboration with Robin Harris and Christopher Timmerman. They were in London and they work on psychedelics. So they were at the Psychedelic Institute and they have this idea, Robin and Carl Friston, they proposed this idea that psychedelics basically do exactly this. So they basically messed up in a predictive coding framework. If you take psychedelics, they basically messed up your model of the world. They relax the precision of the prior. So if you are somewhere here, then you take psychedelics, then your prior got loose. There is less precision in the prior. And in theory, this should generate an increase of forward wave and a decrease of backward wave. And so what they did, they share with us some EEG data in which they provide DMT to the participants. And DMT is a very powerful psychedelic drug, which is used, for example, in ayahuasca, so for the shamanic ritual. So it's a serotonergic drug that generates a altered state of consciousness and visual hallucination. And so basically participants came twice to the lab in London. The first time they got placebo, the second time they got DMT. So on the left, you can see the pre and post injection. For dashed line is placebo, the solid line is DMT. The upper part in blue is the forward waves, and the lower part is the backward wave. And you can see that for the placebo, so I should mention that participants were sitting quietly, closed eyes, enjoying the moment. So during the first session with placebo, there are no forward wave and a lot of backward wave. This is again very much in line with what Cao Yang found in our first study. So no visual stimulation, top-down waves, very nice. The second time they came, they got DMT, and as you can see, as soon as they got the DMT, on the right part, you can see a more precise temporal resolution. So after 5 minutes, they got the DMT, and there is an increase of forward wave and a decrease of backward wave. Again, very much in line with the prediction of our model. So there is an increase of forward wave, even though they had the eyes closed, supposedly, if we want to push the predictive coding interpretation, because their prior was somehow loose and messed up, and this generates an increase of bottom-up prediction error. Okay, we can also test, we also did the other way. So with the DMT, we were on this side. How about the right side? And this was a working collaboration with Dario, actually, Gario Gordillo, and Michael Herzog here in Lausanne. There is this hypothesis that in schizophrenia, maybe we are actually in this right world condition. So schizophrenia patients, they actually may have higher level prior that are much more precise than the control population, let's say. And to be a bit more precise, the story is a bit more complicated than this. I'm referring to Sturter and Corlett studies, in which they propose that that's true for the high level prior. So in green, you can see the patient, the schizophrenia patient, supposedly, so this is the hypothesis, where high level prior, they are much more precise in coding of the word somehow, where sensory prior or lower level prior, they are much more distributed, simplifying a bit. So in theory, if we test schizophrenia patient on a resting state or a higher task, let's say higher cognitive higher task, we should see more top-down waves. Whereas if we test them on a sensory or a visual task, they should have more bottom-up alpha waves. So that's what we found. I will focus on the right part. So on the x-axis, you see the frequency. On the y-axis, the power of the wave in decibel. The solid line is the control, and the dashed line are the patient for the forward upper part, and in red, the backward wave. And on the right part, I basically average the frequency band together, and you can see that yeah, you don't see my mouse, sorry. You can, you do. Yes. So you can see that the alpha, in the alpha band, there is an increase of top-down wave in the patient, the dashed line, top-down, and a decrease of the alpha wave propagating forward. So this, again, very much in line somehow with the prediction of our model, that there is an increase of top-down wave during resting state and a decrease of bottom-up waves in the forward waves. Yes, so they also had the same participant doing a visual task, a vernier visual task. We don't care too much about the specific of the task. They were just actually performing some sensory data. And what we found here, again, here there are all the frequency band. I'll focus on the alpha band. 0 is the onset of the stimulus. And again, the dashed line are the patients. So you can see that as soon as the stimulus kicks in, so the onset of the stimulus, there is an increase of alpha wave specifically for the alpha wave for the patient compared to the control. And this effect was specific to the alpha. We didn't see it in any other frequency band. Once again, somehow in line with the prediction of our model. Okay, so I basically introduced to you the idea that this alpha traveling wave may be related to predictive coding processes. And that's the main hypothesis that we are trying to test currently in the lab. And of course, the two studies that I show, both the DMT and the schizophrenia one, are very indirect, because in both cases, you have to commit to the idea that the DMT alter the prior, or vice versa, then in schizophrenia, they have stronger prior. So it's very interesting, goes in the good direction, but it's very indirect. So what we did now is trying to test this a bit more directly. So we try to manipulate the prior experimentally to see whether we can actually manipulate the waves as well. And so I'm going to present very quickly ongoing works. This is the work of Martina Pasqualetti, who is a PhD student in the lab. And she's having fun basically studying participants with a statistical learning design. So there is a fixation cross, which is actually below the shapes. And the shapes are changing every second. And the participants are instructed to press a button as soon as they see a target shape, which is presented at the beginning of the trial. So as soon as they see a triangle, they should be pressed as fast as possible. And what we record is the behavior, of course, reaction time, and scores. I will mention this in a second, pupil size, and of course, EEG traveling wave. Okay, so very easy statistical learning task, detection task, they have to detect the shapes. What is interesting, we manipulate this transition. So how the shape changed over time, what we did basically, we have 5 different conditions. In one condition, the change of the shape, the transition between shapes is very predictable. So for example, the circle very often became a square, and very unlikely the triangle and the diamond. So in this case, it's very predictable to see somehow the transition between the shapes. And to the other extreme, it's totally unpredictable. So any shape could become basically any other shapes. So we manipulate this predictability across transitions. And our hypothesis, we have 2 hypotheses. The first one is that, well, if backward wave actually tracks prediction, we should see more backward waves in the more predictable case, so on the left, and we should see more bottom-up forward waves, or at least less backward wave in the less predictable case, all the way to the right. This is our first hypothesis. The second one is that in each condition, we have an expected case. So let's take the 90% case. So the square can become a diamond. This is expected, but sometime it will become something else, either a triangle or a circle. And this is unexpected. So this is a prediction error somehow. So in theory, we should see an increase of forward waves during an unexpected event. Yes, following an unexpected event, we should see an increase of forward waves. Okay, so we have these 2 hypotheses to try to test directly this link between traveling wave and predictive coding. So what Martina did is split, first of all, the participants into groups based on whether they use the statistics or not. So we basically have the reaction time, and we compare the reaction time when the shapes was expected. So in this case, when it's very predictable and it was a target, then we can see how much they are faster compared to when it was unpredictable. So if this difference is big, it means that they are using the shapes because they are basically anticipating the target because they know it's predictable. If the difference between rare and expected is basically 0 or negative, then they are not using the statistics at all. So we are basically splitting participants in learner and non-learner, even though it's not really learning, it's more about using the statistics or not using the statistics. And I forgot to mention that actually we ask at the end of each block to judge the reliability of the sequence. So we ask them explicitly at the end of each block, how much could you track, how much could you predict the statistics? And they were actually able, you can see it here, on the x-axis are the 5 conditions, on the y-axis the confidence level, and you can see that all of them, even the participants that learn less, so they use less the statistics in red, still they judge as more confidence the more predictable condition. So they are consciously aware of the transition of these statistics. And even more for the participant who use more the statistics. So we are basically splitting, showing the confidence level for the 2 populations, the one who use more and use less the statistics, and both of them show that they are aware of these statistics. On the right, you see the reaction time for the different transition probability. And again, you can see that, especially for the green one, for the group that use the statistics frequently, they are much faster when it's expected, which of course kind of makes sense because that's how we define them. So the more expected is the shape, the faster they are in responding. So from a behavioral point of view, we see that the 2 groups, they learn the statistics, one group more than the other, of course, and they're aware of this transition over time. So what about our traveling wave? So first of all, we see overall how the traveling wave behave over time. 0 is the onset of the shape. And we have on the left part the backward wave, on the right part the forward wave. You can see that before the onset of the shape, there is an increase of backward wave irrespective of the condition. And there is an increase of forward wave after the onset of the shape. So once again, kind of suggesting that backward waves are somewhat related to expectation, and forward wave are somewhat related to sensory processes. And this is actually entrained over time. So we're actually now trying to investigate whether this entrainment is a real thing or not. So there is a temporal dynamic of the waves, but more importantly, we test the first hypothesis, whether sequence predictability influence our backward and forward wave. And the answer is no. There is basically no effect of the condition of our forward and backward wave. So on the left side, you see the backward waves as a function of the condition. So 2 to 5, we exclude the first one because it's completely unpredictable. For the two groups, in red, the one that use the statistics, in green, the one that don't use the statistics. Either way, there is no manipulation, there is no effect of the condition of our backward wave or our forward wave. So basically, we have a negative results for the first hypothesis. If you manipulate the predictability in statistical learning, backward wave or forward wave, they couldn't care less. There is no manipulation of the wave based on this manipulation of the probabilities. But we found something on the second hypothesis. So here we are basically comparing the difference between forward and backward wave between rare and expected. So in a predictable case, sometimes there is an unexpected event. And in this case, you can see here on the, now I change a bit the thing on the left panel, there are the participants that use more the statistics. On the right part, there are the participants that use less the statistics. In red, you have the rare event, and in blue, you have the expected one. And you can see that for the rare event, there is an increase of forward waves that increases as a function of the condition for both groups. So both for the more and for the less. So even if they don't use the statistics, there are more forward waves after an unexpected or a prediction error, so after an unexpected event, which is kind of interesting because it shows that still these forward waves are somewhat related to somehow prediction error, even though we are still investigating, we are trying to correlate whether this is reflecting, for example, an increase in arousal. So we are using the pupil to see whether this is actually the case. And here is, the same thing, basically. So the difference between expected and rare, and you can see that for all the condition is higher than 0. I mean, not for the first condition, but for the other three is actually higher than 0, meaning that there is a difference between rare and unexpected. Okay, so this is still work in progress somehow, but seem to suggest that indeed traveling wave may be tracking, especially the forward one, some prediction error effect, but not the top down, the backward one. All right, I want to show the very last, again, very preliminary data, and this is in collaboration with Matthew Nassar at Brown University. He had a very cool task in which he's basically asking subject to intercept a cannonball. So there is a cannon in the center of the screen, and this cannon is shooting balls, and the participant has to put a shield to intercept the ball before it hits a target, let's say. And so the participant first places the shield, the center of the shield, then the cannon shoots, and so you can see where the ball is landing, and then the participants see the extent of the shield, which changes at every block. And the idea is that here they are disentangling when the shooting of the ball and the fact whether the target was successfully intercepted or not. Now, there are two conditions in this case. In the first condition, the oddball condition, the cannon keeps shooting more or less in the same direction. So let's say it keeps shooting vertically with some variance, of course, and this is constant across the blocks. Sometimes there is an oddball condition in which the ball ends very far from the mean of the distribution, but the subject is instructed to ignore this condition because it's an oddball. So they have to ignore it. They keep track that, okay, the cannon keeps shooting one direction, sometime we'll shoot somewhere else, but please ignore it because the cannon will keep shooting in the right direction. In the other condition, the change point condition, the cannon keeps shooting one direction, but then it will change. So now the oddball is actually a cue that you have to update your distribution. You will have to follow this new direction because the cannon will keep shooting in the new direction. So in one case, you have the oddball condition. Please ignore the oddball because it happened only once. In the second condition, the change point, you have to update your model of where the cannon is shooting because now the cannon will keep shooting in the new direction. And what we want to see is whether traveling wave reflect this update somehow. You have to update your model in the change point because you need to update the distribution of where the cannon is shooting. but not in the other case. We want to see whether there's a difference in traveling wave in the oddball and in the change point, knowing that the only difference is in the update of the model. And quite surprisingly, we found a very nice difference. So on the left part, so on the upper part, you see the forward. On the lower part, you see the backward waves. The first column is the oddball. In the solid line is the oddball trial. So sorry, the first column is the oddball condition. The second one is the change point condition. The solid line is the oddball trial or the change point trial. And the dashed line is all the other trial when nothing happened. And what we found quite interestingly is that only during the change point condition, so when there is a change point and participant has to update the position of the model, then we see an increase of forward wave, as if these waves are somehow carrying on the prediction error that is saying to the higher region, wake up, there is something happening, we need to update our model. And this was very specific only to the change point and not to the oddball condition. So if we compare specifically the oddball trial for the oddball condition and the change point condition, we see a difference specifically in the change point. And more interestingly, there is also a very nice correlation between the extent of the update, so how much they need to update their position, so the prediction error and the update, with both forward and backward wave as a function of time here. So it seems that indeed this traveling wave in this specific task are somehow related to this update of the model. All right, so to sum it up, the first main take-home message that I like to push forward is that oscillations are amazing, but they are super important. It's super important to consider not only the temporal component, but also the spatial one. So how they propagate as traveling wave. In respective of the predictive coding interpretation, it's still important to consider oscillation as wave. So taking into account the spatial component as well. Then what I show basically is that forward waves are related to visual stimulation, and backward waves somehow related to inhibition, and I didn't show it here, attention modulation, but we have a study in which we show that visual attention modulates backward waves. I also show that traveling waves are related to both psychedelics and schizophrenia, so there is, psychedelics do modulate traveling wave, and in schizophrenia patients, we have a difference in traveling wave. I like to think that there is this predictive coding interpretation. This is what we are trying to test now more explicitly. But even without committing directly to this framework, there is still a difference between all these conditions as shown by the traveling wave. So what we are doing now in the lab, we are having fun with different tasks, different models, so binocular rivalry, working memory, and computational models to try to understand a bit deeper and more precisely the link between traveling wave and predictive coding mechanisms. So I'd like to thank all the collaborators, my amazing OC Pred team, Leslie, Jacob, Antoine, and Martina, and all of you for your attention.
Yeah.
So, I'm a little curious about the assumptions about your forward and backward traveling waves in terms of across these midline electrodes and the assumption that it's the same oscillation and therefore the same phenomenon. Can you dive into that a little bit?
Yes, that's a very good point.
Okay.
So, yeah,
Repeat
sure.
the
Sorry.
question.
Yes. So the question is whether across the electrodes, if you understand, across the midline of electrodes, whether it's the same alpha oscillation that we see propagating or whether there are different sources, for example, right? And that's an excellent point. So we don't know, basically. Based on EEG, we can't really say, because it could be indeed that there is this famous paper from Alexander Zigalov and Ole Jensen, in which they show that two dipoles are actually enough somehow to show in the perceptual vehicle, so it was very specific, but still could be enough to show whether this is the case. And it might be, with EEG, I think we are fairly limited. There are other studies that show that there are these traveling wave propagating, even at the macro scale with intracortical patch on animal, on monkeys notably. And but with EEG, we are fairly limited. We are using different methods, like considering more electrodes and fitting a plane, for example, or considering only splitting in two the number of electrodes posteriorly, centrally, maybe actually in three, and frontally, to see whether we can see the phase fit across the region, to see whether there is a gradient which is truly true. Because if we see it only occipitally, we could see it with the 2D-FFT, we could see a wave where there is only occipital and not frontally, and we would still see a wave. So we try to split the analysis more locally, let's say, and we still find the same results. It could still be due by the dipole, and with DEG, we are still fairly limited in how much we can infer. So we could do source localization, for example, or what we plan to do on the long run is maybe use other methods like MEG or something that is a bit more spatially resolute. But it's a very valid point. We don't really know, basically. Yeah.
Maybe you already partially answered, but there are some bunch of questions also from people online. And they asked if the role of the different brain rhythms, okay, in your predictive coding, let's say, and also some of them suggest also to use source reconstruction methods in order to analyze this kind of phenomenon. So if you can comment.
Makes sense. That's a very good point. So yes, source localization makes a lot of sense. We actually plan to do something like this, even though I'm more I like more to use other like MEG or intracortical recording in epileptic patient, for example. There would be a more direct way to test this. And about other rhythms, it's true that there are this influential work from Bastos, for example, in which it suggests that higher frequency like gamma maybe are related to prediction error, and lower and alpha and beta maybe are related to prediction, which is true, but it's unlikely that gamma propagates with EEG. It's unlikely to see a propagation of gamma at the macro level, so between region. So I'm a bit more skeptic to look at gamma with EEG, but maybe with other methods like MEG or intracortical recording, that would make sense to broaden a bit more the frequencies.
Okay. So thank you. Okay, so I will present our next speaker. So Isotta Rigoni is a biomedical engineer. She completed her PhD at Aston University in the United Kingdom, where she used multimodal electromyography and electroencephalographic acquisitions to investigate brain-to-muscle connectivity following muscle stimulation for postural control application. She moved to Serge Villemo lab at the beginning of 2021 to work on EEG network analysis for epilepsy across vigilance states in both humans and animal models, and expanded them to the investigation of structural functional coupling for surgical outcome prediction. Since October, she is meta assistant, congrats, at the University of Geneva, working on the impairment of memory consolidation during sleep in patients with temporal lobe epilepsy. I will leave the stage to Isotta.
Thank you. Hello. So as you understood, maybe I will talk about EEG brain networks and how we can use them in the context of epilepsy. So first of all, a quick recap on EEG, what David already told us. EEG can directly measure neural activity. It's convenient because it's cost effective, it's portable, it's non-invasive, and can come in different formats because we can have low and high density EEG coverage. And then what we mentioned at the end, what Andrea mentioned at the end of his talk, we can use electrical source images to reconstruct the activity in the brain that gives origin to the measures, to the potential that we measure at the scalp level. And why can this tool be useful for epilepsy? So first of all, epilepsy is a neurological disorder that is characterized by recurrent unprovoked seizures. But on top of seizure, another classical feature of epileptic patients is the occurrence of interictal activity. So the occurrence of spikes that are asymptomatic. So the patients do not perceive them, they don't have symptoms, but they are able to disrupt cognition if they occur, for example, during memorization or retrieval of a memory. Spikes are important because they are often used, always used in absence of seizure, especially during the pre-surgical routine. Basically, what we do is we do source localization on spikes to image the irritative network, which is the network that is overlapped to the epileptogenic network, which is the network that in case medicines don't work, in case medications don't work, needs to be resected to achieve seizure freedom. I've talked about networks because epilepsy is nowadays recognized as a network disorder, while once it was thought to be a disorder linked to one specific brain region, different of course from patient to patient. Now it's more and more recognized as brain network disorder. And why EEG then can be useful for this? Because with EEG we can image different rhythms, seizures if we're lucky, spikes if we're less lucky, or nothing at all if during the recording that usually lasts 20-30 minutes, there's nothing, there's no epileptic seizure. So there's no epileptic sign. So why EEG? Because with EEG we can extract biomarkers of diagnosis. So in absence of spikes, we should be able to tell whether, we hope that we're able to tell that EEG, even in absence of spike, belongs to an epileptic patient rather than an healthy control. And for prognosis, because it's important to be able to predict whether the surgery can yield seizure freedom or not in a patient. Here you have some examples of clinical recordings. On the left, you have a focal interictal spike. That's exactly the interictal activity that I was talking about. As you see, it's very brief. And in this case, it's focal because it's only occurring in a bunch of electrodes in the left temporal area. While on the right, you see very widespread discharge that is a generalized seizure. It's called generalized because it's widespread across all electrodes and it's occurring across both hemispheres. So electrical source imaging, ESI, can allow us to go from the scalp to the sources. How? This is just a snapshot of the pipeline that we use for performing electrical source imaging, where we start from the functional signal that we want to reconstruct, so EEG recorded either with high density or low density, and we couple it with the anatomical data that we use to model the conductivity in the brain across the different layers, the brain, the skull, and the scalp. And when we put these two things together, we are able to solve the inverse problem. So we're able to reconstruct the activity at the source level. And then with the use of an atlas, we can group this activity and summarize this activity across different brain regions and extract ROI time series. So if you are not familiar with this and you want to know more, this is a link to a tutorial that me and my colleague Nicolas Roeri did also together with David and Maria in the context of a Synergia summer school 3 years ago. So if you want to take a look, go for it. Once we have reconstructed the brain regions, the time courses of the brain regions, what can we do with these time courses? If we're interested in understanding how these brain regions work together at the same time or at the same frequency, or how they cause each other's activity, then we can look at connectivity. Depending on whether we're interested at the level of synchronicity in time, in frequency, or whether they're causing each other according to Granter's causality, then we can choose different type of connectivity metrics. And once we have measured the statistical dependencies between the two brain regions, then we can reconstruct the brain network. In this case, it's a functional brain network because we're measuring the level of synchronicity between the activity of two brain regions. And simply in this brain network, the nodes are the brain regions that we reconstructed with the atlas, and the edges are is the strength of the functional connection between these two regions. And on this brain network, what we can extract are measures that describe the topology of the network. So measures that describe integration or segregation, for example. I will quickly mention what they are because we will see them later. So the integration of the network reflects the ability of the network to rapidly combine this information from, I don't know why that is like that. Sorry, it shouldn't be. But, so integration is the ability of the network to rapidly combine information from distributed regions. And this is reflected by the global efficiency, for example. The segregation instead of a node, and when we average it over the network of the whole network, reflects the tendency of the node to cluster, to create strong clusters or modules across the brain. So it reflects the level of functional spatialization of each of these clusters. And this measure can, of course, be calculated at the whole brain or at the nodal level. So now the question is whether these measures, this approach can be useful at all for epilepsy. As we said before, we're interested in the diagnosis and the prognostic of the surgery. So in terms of diagnostic value of connectivity, what we showed in the lab was that even when we look at EEGs that don't have any spikes, we're able to tell the difference between a patient and an healthy control just by looking at the average level of integration of the functional network, showing that patients indeed have higher integration or higher global efficiency than controls. This was confirmed later on, and in another group of patients, where I showed that not only patients have higher connectivity, higher integration, but that this occurs at specific frequency bands, which are the low frequency bands. And that occurs better during wakefulness than sleep, probably because during wake we have the background activity that is higher than during sleep and covers spikes that are not detected at the scalp level. For the prognostic value of connectivity, is there any? So when they looked at the added value for the localization of seizure onset zone in patients who were seizure-free, meaning that we are sure that the zone that was resected was the correct zone. They showed that indeed, when we only look at the source with the maximum power, so the reconstructed source with maximum power, we only reconstruct 30% of the seizure in the actually resected zone. Instead, when we look at the outflow of the nodes, so when we look at the source with the biggest outflow, and the outflow is basically a network measure that reflects how much that single node affects the other nodes of the network, then the percentage of seizure reconstructed correctly in the resected area is more than doubled. Otherwise, like in another study, what they showed, they looked at spikes of patients who were seizure-free or non-seizure-free, and they showed that the patients who were not seizure-free indeed had a higher global efficiency, functional global integration than patients with good outcome, suggesting that probably during the spike, which is a proxy, of course, for seizure, is not a seizure, but it's still a pathological discharge, suggesting that the level of integration or propagation of the interictal network was harder to disrupt by simply resecting the area. And so this, the fact that connectivity can indeed be useful for epilepsy motivated us to look at it from a slightly different perspective, which is the one of graph signal processing. Graph signal processing is a pipeline that has not been invented by me, that has been around for some time. It was mainly used it is used for coupling the structural signal and the functional signal, but was mainly used with fMRI. And only recently it is now being applied to the EEG. Before we dive into it, we need to make sure that these two concepts, the graph and the graph signals, are clear. So a graph is an entity made of nodes and edges, where edges connect pair of nodes. A graph signal is instead a signal that lives on a graph, that changes over time. As you can see, the entity here over time is the same, but the value of each node changes over time. So while this one does not change, the activity of each node can change. So in the context of brain data, what is a graph and what is a graph signal? The graph is the structural connectome. So it's our entity on which the signal is defined. The structural connectome is a graph made of nodes and edges, where nodes are the region of interest and the edges are the white matter fibers that connect this region of interest. And the graph signal, what can it be? Is of course the signal that is defined on this graph. So the functional brain signal. How can we measure it? Either with fMRI or with EEG or with MEG, with whatever you want. In our case, it was with EEG. Of course, reconstructed from electrodes to the brain area. And as we can see here, we have activity of each node, so of each ROI, that changes over time over the structural graph. So in practice, it's very quick. How do we obtain the structural connectome? From a T1 weighted MRI, we can segment it and obtain brain areas with the use of an atlas. With diffusion weighted imaging and tractography, we can obtain the white matter tracks. So when we put it together, we obtain the structural connectome. For the functional signal, as we said before, EEG plus forward model describing the conductivity in the head, we can reconstruct the brain activity at each time point, and that's our graph signal that lives on this structural graph. Okay, quick recap about the Fourier, so that we understand what we're doing with the graph signal processing. So in the time domain, we use the Fourier transform every time. Let's think about just a one-dimensional signal that is defined over time. You can see it's made of a superimposition of two frequencies. We know that this signal can be decomposed into sine and cosine waves, right? The Fourier modes. Where do these waves come from? We'll see it in a bit. Bear with me. They come from the decomposition of the time graph. And then, of course, we know that this signal can be reconstructed as a linear combination of these modes, where we filter the signal. We want to remove, I don't know, the 50 Hertz component. We just put the coefficient of the F50 to 0. Okay, so what happens in the graph domain? In the graph domain, Now the signal is not defined over time, it's defined over a graph. So if we decompose the graph over which the signal is defined, what do we obtain? We obtain the Fourier modes that we can use to reconstruct the signal as a linear combination of these modes. These four modes here, those that we are mostly used to, are exactly the same respective things of what we see in the graph signal processing here. They are spatial maps. So again, we just said that we can go from time to frequency with the Fourier transform and backward with the inverse Fourier transform. We use it every time to filter our data. Where do these Fourier modes come from? As we said before, they are the eigenvectors of the graph Laplacian of the time graph. What is a time graph? The time graph is just a one-dimensional ring graph where the first and the last point are connected. So it's just the graph over which our signal is defined. When we perform the graph Laplacian of this graph, we obtain time frequencies. We obtain frequencies of 1, of 2, of 3, of 4, which are the normal frequencies that we use when we filter our signals, and we obtain the harmonics, which are the time waves. So how does it work in the graph domain? In the graph domain, we can graph Fourier transform the signal. So now the EEG is defined on the structural connectome rather than in time. And if we decompose the structural connectome with the graph Laplacian and if we decompose the graph Laplacian of the structural connectome, then we can obtain the network harmonics, which are the spatial maps that are the eigenvectors of this graph Laplacian. What characteristic do have this eigenvector? They are ordered by eigenvalue, which in the case of the time domain is just the frequency, 1 Hertz, 2 Hertz, 50 Hertz. In this case, they're spatial frequencies. So these eigenvectors, as you can see, they're just spatial maps capturing different connectivity patterns, structural connectivity patterns on the cortex. And while you can see that the first ones are very smooth on the cortex, so they capture the long range connectivity tracts of the structural connectome, while the last ones are more coarse, meaning that while the frequency, the spatial frequency increases, the smoothness on the connectome decreases. And these are spatial frequencies rather than time frequencies. So how can we do this? How can we perform the graph Fourier transform? Exactly the same way we would perform the Fourier transform on a time signal. We would just multiply our network harmonics by the signal of the ROI in time. And what we obtain here, we call it collector spectrum, which is just the frequency spectrum that we would have with a Fourier transform. And of course, we can go the other way around, so we can reconstruct the ROI signal as a linear combination of our harmonics. In other words, that's what we can do. We can write the brain activity at each time point as a linear combination of these harmonics. So how do we do the filtering? In the EEG, we have some frequency bands, right, of interest. We just select which coefficient to retain and which coefficient to put to 0. In this case, we don't have frequency of interest because these spatial frequencies are defined over space and they're not yet associated to specific cognitive mechanism. Of course, it comes from a structural connectivity. So what was proposed was to split the connectome spectrum into two, what we call the low frequency harmonics and the high frequency harmonics. So the first part of the spectrum and the second part of the spectrum, where the first part of the spectrum is characterized by these very smooth harmonics that are smooth spatial maps that, because of their smoothness, capture integrative patterns, because integration is about combination of distributed activity across the brain. While on the other side of the spectrum, we have coarse spatial maps. They are more, as you see, they capture clusters exactly as segregation, what segregation reflects, right? So what can we do with it? We can filter. And how do we filter the EEG? We can filter the EEG using a subset of either one or the other harmonics. So if we want to reconstruct the integrated part of the signal, we use the low frequency harmonics. So we just reconstruct brain activity at every time, only using the first set of harmonics, as you would use alpha band coefficient to reconstruct alpha activity. And we do the other, we reconstruct it with high frequency harmonics only if we want the more segregated part of the signal or decoupled from the structure. We call about coupled and decoupled because the integrated portion of the signal follows the main axis of communications of the brain, frontal to anterior, left to right. And so in that sense, we say that it is more coupled to the structure. But of course, these harmonics all come from the structural connectome. And then what we can do is we can measure the structural decoupling index for each region. So for each region, we reconstruct the portion of the signal that is coupled, the portion of the signal that is decoupled from the structure. The ratio between these two gives us an idea of whether the signal of this region is more in the low frequency harmonic spectrum or more in the high frequency harmonic spectrum. And now, can we apply this to epilepsy? So can GSP predict the surgical outcome? So in this first study, we looked at 9 patients with right temporal lobe epilepsy and 8 patients with left temporal lobe epilepsy. All of them had high-density EEG recording, spike marked, and we performed graph signal processing on the spike using, and of course, this is the limitation that we're trying to address now, using a consensus structural connectome from healthy controls. And what we were able to show is that during the spike, so this is the time course of the spike. In vertical, you see the start, the half rise, and the peak of the spike. And what I show here is a signal measure at the whole brain level, so it's not a specific region, of the energy that is allocated in the high frequency harmonic and in the low frequency harmonic. And what we see is that right before the spike, there's a prevalence of high frequency harmonic, so much more segregated activation, which could be linked to the fact that spikes originate from limited regions, but then during the spike and at the peak especially, we have a significant increase of integration of the low frequency harmonics, suggesting the propagation of the spike to the whole irritative network. And when we look in space, so rather than whole brain, we look at the structural decoupling index of each region, we see, but we can just look at this part, is the most conservative. We can see that for both the left temporal lobe epilepsy and the right temporal lobe epilepsy, it is indeed the regions that are located in the mesial ipsilateral hemisphere that are more coupled to the structure during the spike, suggesting that, I mean, these are the regions, the hippocampus, the parahippocampal gyres, internal cortex. These are the regions that are thought in most of these patients to be where the seizures are located, because these are all mesial temporal lobe patients. So it makes sense that it is indeed these regions that to the structure during the spike than others. And this pipeline was applied to a bigger data set in what is Louise de Vouter-Doplincler PhD study. She basically expanded the study to 33 patients who were all operated and for whom we have surgical outcome at 1 year. And she was able to replicate the results. So during the spike, we see again before spike and at the peak integration. But then interestingly, when we look at the coupling at the peak, we see that it nicely separates the two population of patients who are seizure-free and patients who are not seizure-free, suggesting that indeed when the coupling to the structure is higher, it's easier to disconnect it with the surgery. And finally, since this symposium was about brain waves and oscillations, I just wanted to give you an idea of how the GSP can be used to study spatial and time frequency at the same time. This is a study from an old collaborator, Jean Rouet-Querolt and Valentina Mancini. Basically, they came up with this joint time vertex connectome spectral analysis, and they applied it to visual evoked potentials. That it's just like circles that change velocity over time. Velocity needs to be detected. This task is known to create that effect that you see over here. So inhibition in the alpha and low beta band, and high activation and activation in the high gamma. So this is just the frequency plot, the time frequency plot. That's what it was expected. But they use this framework to study whether different information processing mechanisms, so integration versus segregation, occurs at different frequency bands, at different time frequency bands. And this was done with using the broadcasting index, which is an index that is very similar to the structural decoupling index, only that when SDI is decoupling over coupling, so it's a ratio, the broadcasting index is just the difference between decoupling and coupling, which means that when the broadcasting index is more than 0, the high frequency harmonics prevail, so the content of high frequency harmonics in the functional signal is more represented. While the broadcasting index is less than 0, the low frequency harmonics prevails. And that's what happens when they plot the broadcasting index over time and frequency. So it's exactly the same axis that we see here. Now the broadcasting index we see here at the around gamma, we see that is bigger than 0, suggesting that segregative patterns, because that's what it means. It means that the high frequency harmonics are more represented in the signal, occurs mostly at the high frequency, at the high gamma or high frequency oscillations. And down here, what we see around alpha and theta alpha and beta band, we see that there is, the broadcasting index is much lower, is below 0, meaning that the high frequency harmonics, spatial harmonics are more represented in the signal. So altogether, what they were able to demonstrate was that indeed localized electrical activity occurs mostly at high temporal frequencies, while spatial distributed activity is more likely to occur at low temporal frequencies. These are just some references for the GSP, and thank you.
Okay, right.
Yeah, a quick one. So you presented several methods based on graphic method from the simple global efficiency, which is a single time.
Yeah.
It is more complex decoupling index. Have you ever compared whether, for example, there is an advantage in using one or the other?
Yeah, that's exactly what we're doing. Yes.
Because the question
See, he just asked whether there is an advantage of using purely functional connectivity measure or of using graph signal processing. So I had an extra slide, but I don't have it here. But basically what I wanted to, sorry, I don't know what I did. Is that we are now, so Margherita Carboni showed that high,
go here.
No?
Here, sorry. So we are sure that the
Sorry.
there's in this slide, I think. Wait a
Yeah.
moment. It's this one.
Yeah, exactly.
Okay.
Perfect. Yes. So basically here what she showed was a nice effect of separation between the two with just purely functional connectivity. What we're doing, what we did now, we measured the same thing on our data and we find exactly this effect, which is opposite of what we find with the graph signal processing. Meaning that when we talk about, low frequency and high frequency spatial harmonics probably not necessarily reflect functional and functional integration and segregation only, because they are capturing a different level of network characteristics. And we're trying to, to compare, we're trying to use these measures to understand better the results that we obtain with the graph signal processing, and then we will see which one is more effective. But for now, it looks like that the level of coupling during the peak from the GSP is more informative than the global efficiency. Yeah, exactly. Yeah. Yeah.
I was wondering, because I guess for epilepsy it's relevant to look at these kind of things at the single voxel level instead of putting it into our eyes. And we try to apply this at the voxel level.
Yeah, no. So it's true, that's exactly what they do when they look at the power of the source reconstructed signal. to do the GSP at the voxel level, they don't do it even when they use fMRI, which has a better spatial resolution than EEG. So it would probably it's too noisy to look at voxel level with EEG, with source constructed EEG. Potentially it could be possible, but maybe using a finer parcellation, that yes. Yeah.
Perfect. So I will present the next speaker. The next speaker is one of my colleagues. So Edoardo Passarotto is a research assistant at the Department of Neuroscience at the University of Padua in Italy. He holds a Master Science degree in Music, Mind and Brain from Goldsmith University of London and a PhD in Systems Neuroscience from the Center for System Neuroscience and the Institute for Music Physiology and Musician Medicine in Hanover in Germany. So his primary research interests include motor learning and movement disorders in musician and non-musician. He's also a musician too. He's currently investigating body representation in patients during rehab. So I leave this.
Good morning, everybody, and thank you very much for the kind introduction. So you all noticed that our world is constantly changing, right? So whenever we need to do an action within the environment, we need to adapt our behavior to the environment itself, right? So let's consider a very simple example. We want to navigate our house. We want to move in our bedroom in complete darkness. And of course, in this case, we will probably move slower compared to the normal lighting condition, right? Otherwise, we are at risk of, I don't know, hitting the bed, hitting the sofa. And pain will teach us that we need to actually modulate the motor behavior to the environmental condition, right? And this is a very simple paradigm to explain you what sensory motor adaptation means. So basically, we adapt our motor behavior to the environment on a trial and error basis. And sensory motor adaptation occurs at three main stages. The first one is when we collect data from our senses, right? And then we integrate this information in body representation, which are basically a virtual copy of our body based on the sensory information. And then we use this body representation to create a movement, right? To select the appropriate movement through internal models. When discussing body representation, then we need to consider two core concepts. And the first one is body schema. Body schema is simply defined as the interaction between our body and the environment. So basically is what allows us to move in the environment having a more or less aware idea of our height, of our size and our weight, right? So I know that I can move to the right side with one step without hitting any object because I know how long I am, how wide I am, but I know I cannot move forward because I don't fit there, right? On the other side, we have body image, which is a much more vague concept because it's basically it's the representation of our body based on our idea and belief about our body itself. And it's very similar to ask yourself how much you like a specific part of your body or your body as a whole. And therefore, it's very complicated to operationalize body image. According to previous studies, then we also know that body schema is well grounded with visual, tactile, and proprioceptive information. And so it comes from the integration of multiple senses. And so we also can assume that areas in the brain where this multisensory information, when this sensory motor integration occurs, will be involved in body schema processing, right? So like sensory motor cortex and parietal cortex. On the other hand, we know that body image is mostly relying on visual information, right? So how we appear. And because it's extremely vague, it's very difficult to define it. We don't really have any cortical with any neurophysiological correlate for brain for body image. And then we use both. So we use both body schema and body image to create internal models, which we use for sensory motor control, right? And the models are mostly two, the forward model and the inverse model. Forward model is when we perform an action and we create an expectancy of the outcome of the action, right? So we move to the left and I assume I will not hit any objects. On the other side, we have the inverse model when we do the exactly opposite process. So given a desired outcome, given a desired state of the body at the end of the action, we select the optimal action, right? So we select the most appropriate one. And the link between these two models is, of course, feedback. So we decide to do a movement, we create an expectancies. These expectancies might be matched by the outcome of the action. If the outcome does not match the expectancies, then we use the inverse model to modify the motor program in order to achieve the expected outcome. And why is this so important for us? Well, because, think about it, sensory motor integration is extremely important in rehabilitation. So whenever we need a patient to restore or to refine a motor program, we don't only want the participant, the subject, to learn a specific motor program in a specific context, but we want the subject to be able to modulate motor programs, whatever the environment, right? Whatever the specific condition he is dealing with. And so we decided to investigate sensory motor adaptation in a clinical population, in subjects affected by adolescent idiopathic scoliosis. Scoliosis is defined as a spine curvature greater than 10 degrees, measured in Cobb angle, which is the main, I will say, clinical parameter to evaluate scoliosis and spinal curvature. You see an example here on the, do you see the mouse? Yes, you do, perfect. You see here an example of what we mean with Cobb angle. And why idiopathic scoliosis? Well, because idiopathic scoliosis is a musculoskeletal disease, which leads to substantial functional impairments, as well. It can also lead, unfortunately, to significant disfiguration of our patients. So there could be, you know, unfortunately, some appearance issues with these patients. And so we can assume idiopathic scoliosis to lead to substantial alteration in body schema and body image. But not only this, because we are dealing with adolescents, we know that adolescence is a very peculiar stage of life where we undergo significant and substantial changes in our body, which can themselves lead to alteration in body schema and body image. So we ran a first study 2 years ago, where we collected data from 14 adolescents with idiopathic scoliosis, ICE in short, and the same number of match controls, and we assessed cortical activity during simple standing tasks. So basically, we asked participants to stand on a force platform, which was measuring the posture for 60 seconds. And what we noticed is that patients with idiopathic scoliosis showed a substantial asymmetry in the alpha band over central and central parietal regions. And this asymmetry was not noticed in matched controls. We interpreted these results as a possible compensatory strategy to overcome sensory motor dysfunction, which on its turn might be related to body schema alteration. And this was even more evident when altering the difficulty of the task. So when making the task more complicated by asking participants to stand, so to do exactly the same exercise, but with their eyes closed, or producing more postural instability by asking them to raise their hands. And so as you see on the right side of the slide, the asymmetry was even more pronounced. So far, so good. But still, the study design we used, so cross-sectional study design, was not really helpful in draw any causal relationship. So we could not really rule out whether this EG alteration in the alpha rhythm were related to ICE or maybe to the treatment, because we didn't really use any specific inclusion criteria for ICE treatment. And at the same time, we could not rule out whether ICE was affecting internal models, and specifically the inverse model or the forward model, because just consider that even when our participants were closing their eyes and standing upright, they still had information from the vestibular system, so they still had some sort of feedback. And so we developed a new study, a longitudinal one, where we monitor the progress of 20 participants with ACE and also collected data from compatible controls and measure their postural stability, their cortical activity, as well as measure the motor behavior in simple tasks over 3 measurement points at T0, at T1 after 6 months of physiotherapy and bracing, and at T2 after 1 year. Strong point of this study is that at T0, all patients did not have any history of treatment. So basically, those were subjects who were coming to us to join the study directly after the diagnosis of ICE. And just for you to consider, the clinical evaluation of the spinal curvature was performed at T0 through X-ray and only at T2. So we could only know if participants were improving in their spinal curvature on a clinical level at T0 and after 1 year of treatment. And so to answer our first research question, so are EEG alteration in the alpha band related to ACE pathophysiology or its treatment? We used a similar task that we used 2 years before, but in this case, we used a full factorial design where we altered visual information by asking participants to stand for 60 seconds on a forced platform with their eyes either open or closed, but we also altered proprioceptive information by making them stand either on a firm surface or a foam surface. Sorry, it's not a very nice alliteration. So basically on a hard surface or a foam surface. And this was creating a significant postural instability. So on one side, we have the easiest condition. So basically when participants were standing with their eyes open on a hard surface, was the easiest condition, which is also mirrored by the behavioral data the first platform was collecting. So you see there the center of pressure over the 60 seconds of the task is pretty narrow. And then the most complicated condition in our task was when our participants were standing with eyes closed on a foam surface, which was creating a really strong instability, which is mirrored by the behavioral data. You see it here. So basically the sway was much more consistent and substantial. And then we wanted to investigate cortical activity during this task. And we use as a reference EEG correlates of postural control in the literature. And basically what we expected is that task difficulty and task condition difficulty might be mirrored by an increase in theta power over frontal, central, and parietal region, and a decrease in alpha power band over central and parietal regions. And because it is still an ongoing study and we did not collect data from all participants at all measurement points, what we will present you today are just 2 very representative participants, one with a very large clinical improvement and the other one with a mild clinical improvement. This is the data we collected during the most demanding condition in our task. And you can see that the participant was on a clinical level improving quite a lot. So she started from 35 degrees in Cobb angle and went down to 22, which is quite a lot. You see that the stabiliometric data indicates a much strong reduction in body sway and postural instability. And on a neurophysiological level, we see a decrease in theta power and an increase in alpha power, both low and alpha power. And we interpret these results as a facilitation when doing this task. So basically, the participant was finding this task easier, right? So she had to recruit less sensory information. She didn't use compensatory strategy, and the task didn't require her such a high cognitive load. When we actually look at the second subject, which present just a minor clinical improvement, so you see she was moving from 16 degrees in Cobb angle down to 13 degrees in Cobb angle. the situation is actually pretty stable. So the postural data, so basically the center of pressure, didn't change that much between measurement points. And the same can be said about the topography. So theta and alpha bands seem, theta and alpha power bands seem to remain unchanged between measurement points. And our conclusion is that the treatment was actually effective in reducing the clinical parameters, so the spinal curvature was reduced, and this was mirrored on a behavioral level by a milder body sway, so a more confined body sway, as well as the treatment was effective in modulating cortical activity. Regarding our second research question, so is AIS affecting the inverse model, the forward model, both? We designed a task that we thought would emphasize the role of the inverse model. So basically, what we did is we asked the experimenter to point at a target, a specific target on a board which was placed in front of the participant. And the participant was asked to point at the same target, but keeping her eyes closed. So she didn't have any feedback about her performance nor visual information to drive her movements. And we repeated this pointing task for 10 trials for each end and computed the deviation of the touching point compared to the expected target. And what we saw is that patients with idiopathic scoliosis showed a substantially greater horizontal deviation, so horizontal deviation on the horizontal plane compared to healthy controls. And so we analyzed cortical activity during this task. Especially we focused on the alpha power band and the central electrodes in the right and the left hemisphere. And what we found out is that when our participants were moving their right hand, alpha power increase was positively associated with AIS severity. And this was particularly clear when we looked at the left hemisphere, which is contralateral to the movement. And so we come to our conclusion and our preliminary answer. Again, it's a still ongoing study, so we only have preliminary answer to show you. The first answer is to the question, are EEG alteration in the alpha rhythm related to AIS pathophysiology or its treatment? Well, yes, it seems that the treatment is actually related to the clinical outcome, but also to the behavioral, so the postural improvement. It's leading to a postural improvement, and it's also effective in modulating cortical activity in the theta and the alpha band. And regarding the second question, is AIS affecting the inverse model, the forward model, or both? Well, according to our task, the task we designed, it seems that AIS is at least effective in modulating performance controlled by the inverse model. However, we cannot draw any reliable conclusion about the forward model. And with this, I will really thank you for your attention and thank Professor Masiero and my nice collaborators for the work, and of course, Maria and Matilde, who are here with me today. Thank you.
Maybe everything that Matteo can find, for example, the rhythms involved with the correlates, so some alteration involved schema windows representation, you can use the information for some neurofeedback.
This could be an interesting idea. Yes,
Repeat the question.
Yes, so the question is whether these neural correlates we are finding related to body schema might be used as a possible neurofeedback in rehabilitation, I guess. Perfect. Well, yes, possibly it could be an interesting idea. Still, I'm not so sure how practical that could be. So if our participant could really undergo further rehabilitation protocols, because the one they are doing is already quite overwhelming, I would say, in the case of AIS. But more generally, I think it's a reasonable suggestion, a reasonable idea. Thank you.